Hi everyone, this is Mrs. Riddle, and I just want to spend today talking with you about a couple of topics in statistics that we're studying. So, the first thing I want to talk about is this is a little hard to read at the top, but I think it's important to just pause and ask yourself, have you heard statistics before, like maybe 81% of public school students are not satisfied with the food provided, or the president has an approval rating of 72%? And sometimes when we have so much statistical information in the news and around us that we oftentimes can take that for granted. But I think it's important, especially if we're making decisions based on a statistic, that we stop to think about how that statistic was generated. What is the source? How many people did they ask? How did they go about asking their questions in their survey? And are these results reliable? So that's what we're going to be exploring in this first part of our unit on statistics, is really being a critical thinker about statistical information. So the first thing to do is to recognize bias. Bias is when the author of the survey or whoever's collecting the data could knowingly or sometimes unknowingly ask a question in a study that will favor a certain response. So I hope that you did the Desmos activity, and if so, you are going to be exposed to these types of bias. I'm not going to spend a lot of time talking about them. You can access this document, it'll be shared with you, and you can also do the Desmos activity and then learn about these a little bit more and have a chance to experiment with creating this type of bias. So question order, preface, 2 questions in one, biased wording, and a desire to please are all types of bias that can influence how someone answers a question. And like I said, this could be knowingly a survey design, could be created to sway people to answer a certain way, and it might be unknowingly. So in surveys, there are different types of questions. An open question allows the people who are responding, they can say whatever they want. So if I just said, what's your favorite sport? And I asked everyone in our Algebra 2 class, we could have 28 different answers. Now, maybe everybody has a different opinion about their favorite sport. Now, a closed question might say, from this list of 10 sports, which is your favorite? And it would limit them. Like a check sheet or a multiple choice type of question where the people have to only answer from a limited set of responses. There's advantages and disadvantages to both. I hope you thought about that a little bit in the Desmos activity. Open questions, maybe you get better results, but if you have 30 different answers, really hard to generalize. Closed questions can help narrow down a focus of a survey into a manageable set of data that you can analyze, but it might not accurately describe how the people feel that were answering your survey. So definitely pros and cons to open and closed questions. Quantitative, we've already talked about those. Quantitative is a numeric response. Qualitative is a quality or a type. Okay, so this part is new, and this is going to lead us into a kind of a deep dive into our activity from last week. A population would be the entire group that you are interested in studying and making conclusions about. That would be a population. A population sounds really big. It doesn't have to be. If I was interested in my honors algebra 2 class, the population would be everyone in our class. So if, let's say there were 28 people in our class, the population would be 28. The entire population of Colorado, so that would be every citizen in Colorado, or maybe every citizen of Douglas County. That would be a population. You could do the registered voters in Douglas County. That would also be a population. It would be everyone that was included in that group that you're interested in. But we can't always study an entire population. Sometimes it's too big, too overwhelming, too expensive, or too difficult to conduct a statistical analysis of a population. And when that's the case, we would use a sample. And a sample is when you just look at a portion of the population, and we call that a sample. So we could randomly draw names from, if our population was Highlands Ranch High School, maybe we randomly selected 100 people to ask questions in a survey. So that would be the sample, those 100 people we surveyed from the entire school. So we've been hearing a lot in the news about a census. A census is a process of measuring every member of a population, and we're currently undergoing a census right now. The 2020 census is happening. It happens every 10 years. So the last one was in 2010, and we have one this year. This just measures how many people there are, how old they are, and ethnicity, I think, is asked in the census. So the census is measuring every member of a population. So if my population was 7th period algebra 2 and I put out a survey and I got all 28 answers back, then that would be a census. Now a statistic comes from a sample. So going back to that idea of 100 Highlands Ranch high school students, if I were to collect some data from those 100 students that would represent the whole population, that would be called a statistic. So it's important to understand a sample versus a population, and I'm going to show you why here. So I'm going to go back to this problem where we worked on growing cells. So there are these cells, and they are measured, and this data was collected. And they're trying, I think, to determine which type of sugar is more successful in growing that cell. So let's just say that these are both samples. I have a feeling that there's a lot of cells that would be in a sample. And so maybe they just measured these cells. So I'm going to say that these are a sample. This is not every cell on that Petri dish or on that slide. This is a randomly selected sample. Okay, so this is a sample of sugar W and a sample of sugar P. So sometimes it's important to understand how far apart the data are. So for example, when we take a look at this data, the mean and the median are exactly the same for both sets of data. So if we only looked at the mean and median, we might think these sugars are exactly alike. There's no difference. It wouldn't matter if you use sugar W or sugar P. The mean and median are exactly the same. But if we do a deeper dive, then we might realize that there's really more going on than what meets the eye. What we could look at, if the mean and median are the same, or we just want to make better decisions, we could look at what's called the spread. And the spread is the range, the interquartile range, or the standard deviation. Okay, so let's take a look. I've already put this data into my calculator. I want to show you one quick thing too. Stat edit. Here I have the first sugar in L1 and the second sugar in L2. It's really nice how they give you the checksum. So if you're ever working these problems in the future and they tell you the checksum is 760, that's how you can make sure you didn't make a mistake. So I'm going to do L1 first. L1 is the first sugar that was given. I'm going to go to Stat, Calculate, One Variable Statistics. Now pay attention, this says L1, so that's what I want. Calculate. And then I get the sum is 760, so I know this is right. So here are two measures of standard deviation. Now let me tell you what the difference is. The S stands for a sample. This symbol right here is going to stand for the standard deviation of a population. So how I remember is this is kind of a reflected, upside down, flipped around P for population, and here is S for sample. Okay, now I'm not saying that that's very mathematical way to remember that, but that's how I remember. So the sample is 11, or the standard deviation is 11. That measures the spread of the data. Now I can also calculate the range and the IQR. So let's remember that 11.1 is the standard deviation. So if I go to Stat Calc, One Variable Stats, I can now change this to L2. That was my second sugar. So if I go second to L2, enter, enter, enter. Okay, my standard deviation is 19. Okay, so the second sugar the data is much more spread out. It is not consistent. So sugar P has a lot more variability in the data than sugar W. So in this case, if I wanted this particular sugar to be more consistent and maybe more reliable and not have data that ranged wildly from one end to the next, then I would probably pick sugar W. So standard deviation is a way to see what is the spread of the data. Is my data really spread far apart, or is my data points really close together? So I think about this too in current times, like if we were doing a clinical trial with some type of treatment or vaccine for COVID-19, we would want that standard deviation to be as low as possible for how successful it was. We wouldn't want it to be a wide range of results. We would want that standard deviation to be a really small number. So there might be some times when we would want variability to be higher, maybe like in some type of genetic work. But oftentimes we would want standard deviation to be small, and that would mean there's less variability. So I would be going with sugar W. So I hope that helps explain standard deviation and how you calculate it. Remember, the S is for sample when we look at that, and this is for the population, and then you can compare two different things and see how their standard deviations compare and make the best decision. All right, that's it for statistics for this week. I hope you all are doing well, and thanks for watching.